the cell cycle, interphase includes G1 or gap1, S synthesis, and G2 or gap2. During these stages, the cell prepares for change.
Interphase begins with phase G1 or gap 1. During G1, cells grow and synthesize proteins needed for mitosis. These proteins also activate the cell's metabolism and break down carbohydrates and liquids that transform food into energy. Chromosomes inside the cell have not yet replicated, so in G1, each of the chromosomes only contains one DNA molecule.
S phase follows G1. Its major function is to copy the cell's DNA. The name stands for synthesis. By using enzymes, the cell copies the DNA molecules of each chromosome. After S phase completes, chromosomes have two chromatids, mainly each chromosome has two molecules of DNA.
After S phase, a cell enters G2 or gap 2, and it involves another period of growth. The cell synthesizes more proteins and organelles like mitochondria. Mitochondria in this stage grow and divide. The cell also resumes normal functions like transforming food into fuel for the cell to stack up on and for its use in mitosis. G2 is the final step before mitosis begins.
This is prophase. Mitosis begins in prophase. Chromatin condenses to form chromosomes in the nucleus.
Nuclear membranes start to break down.
Centrioles appear and move to the opposite ends, and spindle fibers begins to form.
Nuclear membranes start to break down.
Centrioles appear and move to the opposite ends, and spindle fibers begins to form.
Nuclear membrane starts to break down.
Centrioles appear and move to opposite ends, and spindle fiber begins to form.
Mitosis begins in the prophase. Chromatin condenses to form chromosomes in the nucleus.
Nuclear membranes start to break down.
Centrioles appear and moves to opposite ends, and spindle fiber begins to form.
Hello guys, today we will show you and tell you about metaphase.
This is the beginning of the metaphase, and the spindle fibers are connected to the future chromosomes and attaching it together.
The spindle fibers are getting closer and living up and growing to the center in the same duration.
The chromosomes line up in the center of the cell. It's also attached to the spindle fibers at its centromere. It marks the point where the whole cell will divide and when the nuclear division is completed.
Anaphase is when the cell chromosomes split into two chromatid and prepares for further division.
First, the centromere breaks, splitting the chromosomes into two identical chromatid.
Next, the spindle fibers that come from the centrioles start to shorten and pull the chromatids to the opposite sides of the cell. As the chromatid are being pulled to the opposite sides of the cell also begins to elongate. Once the chromatids reach the end, the next phase starts.
Telophase by Jonathan, Vu, Romeo, and Guillermo. First step to telophase is a new nuclear envelopes formed around the two groups of daughter chromosomes. After this happens, a new nuclei begin to appear. While this happens, the spindle fibers begin to disappear. After all this happens, telophase ends.
The first step in cytokinesis is to define the position at which the contractile ring will form.
Second step of cytokinesis is to assemble the actin filaments that form around the contractile ring. In addition, proteins like myosin also form.
The third step of cytokinesis is the contractile ring shrinks and the cells grow further apart from each other. The actin filament shrinks for the cells to grow apart. The final step at this point of cytokinesis, there are two daughter cells. They have their own organelles.